Hi everyone. I'm here to discuss a new topic that is gluconeogenesis. It's another pathway, metabolic pathway of glucose. Glucose stands for glucose. This is glucose. Neo stands for new and genesis stands for synthesis. So gluconeogenesis simply means that synthesis of new glucose molecules. What is new glucose? This is glucose synthesized from sources which are not carbohydrates. Normally, we used to get a continuous supply of glucose through the diet we take. Our diet are rich in carbohydrates, so these carbohydrates break down into glucose. But in situations like starvation, where we are not taking sufficient food or no food at all, the body need to produce glucose from sources other than carbohydrate. So such a pathway is called gluconeogenesis. Why we need to synthesize new glucose molecules? It is because many vital tissues in our body, like the nervous tissues, the skeletal muscles, the RBCs, etc., they depend only on glucose for their energy needs. They cannot utilize energy produced by fat. So it is very necessary that we maintain a constant blood level of glucose for all these tissues to function well. Synthesis of glucose from non carbohydrate compounds is called gluconeogenesis. The major precursors or substrates or raw materials for gluconeogenesis are lactate, pyruvate, and glucogenic amino acids. Propionate and glycerol are minor precursors. The process occurs mainly in liver and to a minor extent in kidneys. The pathway is almost similar to the reversal of glycolysis, but it's not the exact reversal of glycolysis. The three steps catalyzed by three kinase enzymes, the glucokinase, phosphofructokinase, and pyrokinase. These steps cannot be reversed. So, complete reversal of glycolysis is not possible. Reversal is possible except these three steps. So, gluconeogenesis involves bypassing these enzymes on this step and converting the end product pyruvate into glucose. Normally, during glycolysis, glucose breaks down like this and finally produces pyruvate. Pyruvate then enters into mitochondria, gets converted into acetyl CoA, enters into TCA cycle where acetyl CoA combines with oxaloacetate to form citrate, and TCA cycle continues like this. This is the normal case. But during starvation, the pyruvate formed no more is converted into acetyl CoA, but pyruvate is converted into oxaloacetate. This oxaloacetate can be converted into phosphoenol pyruvate. But the problem is that oxaloacetate cannot as such come out of mitochondria as it is impermeable to the mitochondrial membrane. Now, malate. It is a molecule which can permeate across mitochondrial membrane. So oxaloacetate is converted into malate using the enzyme malate dehydrogenase. Malate comes out. Again, malate dehydrogenase convert it back into oxaloacetate with the help of phosphoenol pyruvate carboxykinase. Oxaloacetate is converted into phosphoenol pyruvate. The reaction occurs with elimination of carbon dioxide and it involves consuming GTP for energy purpose. Actually, ATP is in short, we suffer shortage of ATP during starvation. So the body uses GTP as the source for energy. Once phosphoenol pyruvate is formed, it goes through the reversal of glycolysis and reaches fructose 1,6 bisphosphate. This step is irreversible, which is catalyzed by phosphofructokinase. So we have the reaction in the reverse step, which is catalyzed by fructose 1,6 bisphosphatase. Now, as we see in glycolysis, fructose 6 phosphate is isomerized into glucose 6 phosphate by phosphohexose isomerase enzyme. Again, this step catalyzed by glucokinase is irreversible, so it takes an alternate way with an auxiliary enzyme, glucose 6 phosphatase. So all these enzymes, which are given in pink color, they are auxiliary enzymes which are not found in glycolysis but found only in gluconeogenesis. You can see one of the major sources, pyruvate, converted into oxaloacetate, then into malate. Malate comes out of the mitochondria, again converted back into oxaloacetate, then to phosphoenol pyruvate, which eventually gives glucose. Another major source is lactate. Here the lactate is converted into pyruvate using lactate dehydrogenase enzyme. Then other route is the following route is as explained as earlier. You can see numbers 1, 2, 3, 4 and 5. Actually, in number 1 we have some glucogenic amino acids like alanine, glycine, serine, cysteine, threonine and tryptophan. These molecules can be converted into pyruvate. So all these are called glucogenic amino acids. These glucogenic amino acids can be converted into pyruvate and can give back glucose. Aspartate and asparagine can be converted into oxaloacetate. Asparagine and aspartate enter in this part of the pathway, then ultimately moves on to give glucose. Arginine, glutamate, glutamine, histidine and proline, they are converted into alpha ketoglutarate. Again, it takes these steps until malate. Malate comes out and gives ultimately glucose. Isoleucine, methionine and valine, they are converted into succinyl-CoA, and phenylalanine and tyrosine, they are converted into fumarate. Both this succinyl-CoA and fumarate also convert into malate, oxaloacetate and finally gives glucose. So we can say that the major substrates are pyruvate, lactate, and glucogenic amino acids. Now, significance of gluconeogenesis. It maintains glucose level of the body under anaerobic and fasting conditions. Under anaerobic conditions, most of the energy producing steps in mitochondria will not occur. In that case, gluconeogenesis will commence and ensures the glucose level in blood. So during anaerobic conditions, this step will not take because TCA cycle proceeds only during aerobic conditions. We have already studied that sufficient oxygen should be supplied for TCA cycle to run. So in this case, anaerobic conditions, pyruvate is converted into oxaloacetate, malate, and finally into glucose. Glucose is the only source for energy for muscles under anaerobic conditions that I have already told you, not only skeletal muscles but also nerve cells and RBCs and many other cells like testes, etc. They cannot find any alternate source for energy other than glucose. So it is very important that gluconeogenesis should produce sufficient glucose. During fasting, it supplies glucose and maintains the intermediates of TCA cycle. So through this substrate 5, 4, 3, etc. during gluconeogenesis, they are converted into intermediates of TCA cycle. All these glucogenic amino acids, are converted into intermediates of TCA cycle like 2, 3, 4, and 5. It effectively clears the metabolites like lactate and glycerol. During glycolysis, in last step, we have studied that under anaerobic conditions, pyruvate is converted into lactate by the action of lactate dehydrogenase enzyme. When the condition reverses, lactate is converted back into pyruvate by the same enzyme LDH or lactate dehydrogenase. So the lactate which accumulates in skeletal muscles can cause fatigue, which is an unwanted effect. So this lactate which is causing some problem with the skeletal muscles, that is fatigue, this problematic lactate is taken into liver through blood, and in liver it is converted into our useful molecule, that is glucose. So lactate which produced an unwanted effect is now converted into a useful molecule, that is glucose. Glycerol. See, glycerol is a byproduct of fat metabolism. When fats and oils break down, one of the products formed is glycerol, and this byproduct is also very efficiently used to form glucose. So glycerol is converted into glycerol 3 phosphate and then into dihydroxyacetone phosphate, which combines with glyceraldehyde 3 phosphate to form fructose 1,6 bisphosphate, then into fructose 6 phosphate, glucose 6 phosphate, and finally glucose. So these are some of the significance of gluconeogenesis. One is that it is helping in maintaining a continuous constant glucose level in our body. Then it supplies glucose to the vital tissues like skeletal muscles, nerve cells, etc. under anaerobic conditions. Again, it maintains the intermediates of TCA cycle, and it effectively clears the metabolites like lactate, which may cause unwanted effects, and also glycerol, etc. to useful glucose molecules. So in all these cases, the substrates like amino acids, lactate, pyruvate, which are not carbohydrates, are eventually and effectively converted into glucose, which is a carbohydrate. So this pathway is called gluconeogenesis, which simply means that glucose is synthesized from sources or substrates other than carbohydrates. So that's it about gluconeogenesis. Thank you for your patient hearing. Thank you.